So we're going to review the structure of DNA and talk about how to build a DNA molecule. So from your activities the other day, the things that you want to remember is that DNA stands for deoxyribonucleic acid. It is this molecule here, and this molecule, sections of it are called genes, which are the instructional code for very particular proteins in your body. And these genes are wound together, along with the whole molecule of DNA, into structures called chromosomes that divide during mitosis. Okay? So the importance of DNA is that it is the instructional code. Like if you want to, put something together like a Lego project, you might read the instructions and put the blocks together in certain orders. Well, the DNA is the code for how to put amino acids together to build proteins. So the structure of DNA, we call it a twisted ladder, and that's why you see the screw plus this ladder. The ladder being the outside sugar phosphate groups of the DNA molecule, and the inside rungs being the nitrogenous base pairs. But it's not just a straight ladder with rungs, it's actually twisted, right? So when you twist this ladder, you get this kind of helical shape. That's why they call it a double helix. So one more time, you have the nitrogenous base pairs, adenine, thymine, guanine, and cytosine. You have the 5 ring sugars. That's why it looks like a pentagon, 5 sides, and then a phosphate group attached. These are the outsides of your ladder, right here, 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 and here. And these nitrogenous bases are connected to each other down the middle to make the rungs. So the direction of the molecule goes in 2 different directions. That means they go opposite. One's going one way, the other's going the other way. And that's this 5-foot, 3-foot direction. It indicates how the phosphates and sugars are linked. So again, the important thing is that it's opposite direction. If one side of the ladder is going like this, the other one's going backwards down the other way. The code, the rules of DNA are that adenine A always binds with thymine T, and G-guanosine always binds with cytosine C. Okay? So adenine and guanine have 2 carbon-nitrogen ring structures called purines. Thymine and cytosine have 1 carbon-nitrogen ring structure and are called pyrimidines. And again, A binds with T, G binds with C, and it's basically this complementary shape idea, right? You have to have an opposite shape in order to connect, and A has the opposite shape of T, these 2 bonds, whereas G is able and C are able to form these 3 bonds. Okay, so these complementary shapes. Here's a tricky little way, if you want to remember it, some mnemonic devices to help you figure out permines and pyrimidines. Not that important at this moment. And here's a nice summary of everything that we just went over. And so that's that. So today's activity is about building a DNA molecule. So here are the instructions for doing that. You're going to need 6 base pairs long. Your molecule has to be. That means 6 connected base pairs down the middle. And in order to do that, you're going to need 12 base pairs total. You're going to need 12 deoxyribose sugar pieces and 12 phosphate groups. You're also going to need to color code your model: adenine, yellow, thymine, blue, cytosine, orange, and so forth. The sugar will be purple. And then the second page are your pieces. Now notice you're going to need 12 sugars, but I only have 1. So in order to get all of the pieces you're going to need, you have to right-click and copy the image. And when you do that, it's going to make another copy. Now, I would strongly suggest that you go ahead and color your pieces first. So you can go to your drawing tool and color things in first and then copy them, so you can do less coloring throughout your assignment. There are several links that are going to help you understand DNA better before you try to build your model. One is this one, which gives you a 3-dimensional structure of the DNA. If you twist it around, hopefully you can kind of see that double helix structure. And so what we're looking at here, the red and the gray, are kind of that sugar phosphate backbone twisting around. And then down the middle, you've got your blue pieces, which are your nitrogen bases. And again, if you keep twisting that and moving it, you'll get different angles that you can see more easily. In addition, there's this nice DNA mutation simulation. We haven't really talked much about mutations yet, but you certainly can figure that out, that you can make mistakes in the DNA code and see how that affects the proteins. But even if you just look at the DNA structure, you can see the two hydrogen bonds between T and A, thymine and adenine, and the three hydrogen bonds between guanine and cytosine. You also can look at what happens with RNA, right? So RNA has a little different code. There is no thymine, so every time there's an A, you're going to get a U on this side of it. This is the messenger RNA being made. And then you can do other things like translate and show the protein. The last one I want to show you is another base pairing interactive activity. You can move these different base pairs out to your work area. You can move them around and try to figure out if you can get them to connect to one another in the right orientations. You can show the atoms, so you can try to line up the hydrogen base pairs. And then eventually you can try to join the pairs. If it doesn't work, it's going to let you know. And in the end, if you have to, you can give up, and it'll kind of show you the answers. And then you can move on to other things like adding in the sugars and the phosphate groups and whatnot with that. So this is really helpful to show how those molecules are assembled. And that's it. That's DNA. Have a good day.